assistant professor at Cornell University in Geneva. She's working on controlled environments in agriculture. So these are protective glasses, plastics, netting, and she's interested in biocontrol. She's interested in plant-insect interactions, IPM in general. So Samantha, she completed her PhD in Cornell. So she went back to her institution where she got her PhD with her new position. She did that under Greg Loeb. She finished in 2022. She did her bachelor's and master's at Utah State University. And after that, after completing her PhD, she did a short postdoc at Purdue University working with Laura Ingwall. So welcome, Samantha.
Awesome, thank you. Can everyone see these slides from the back? People on Zoom, if there are folks. I think everything should be checked out, so I'll go ahead and get started. Thank you for the invitation to be here, Cesar. It's my pleasure. This is my first seminar as a new faculty member, so this feels kind of big to me. I'm very happy to be here, and I hope you guys have a good time as well. So my topic today is insect dynamics in controlled environments, really focused on tritrophic interactions with an IPM perspective, but also thinking out of the box and thinking about other forces that are influencing our plants and insects and their interactions, also with some of our natural enemies at the top trophic level. So Cesar gave a really great comprehensive background. It was perfect. So basically, recapping this, I'm from Utah. I got my bachelor's and master's there. I went over to New York, did a PhD, then a postdoc at Purdue, and I just started my new position back in November, so I'm pretty new. But something that wasn't included is some of my favorite foods I've accumulated through all of my different adventures. I don't know if any of you have been to Utah, but the pastrami burger is like the sandwich there. It's amazing. Obviously, I've gained a really great appreciation for bagels living in New York now. That was not a thing out west. No one cares about bagels there. Here, different. And I don't know if you guys have tried this sandwich in Indiana. This is the pork loin sandwich. It's like comically weird. The bun is really small and the pork loin is massive, and it's on purpose. So it embarrasses you as you're trying to eat it because you don't know how to approach it. It's not a great sandwich, but it's worth trying just so you can say you tried it. All right, and back on topic. I thought I'd start this talk off very broad, defining what a controlled environment is, and that's actually a very tricky question. We were talking about definitions during our meeting earlier with Cesar's lab, and it's so hard to nail down definitions for really squishy things. Controlled environment is so dynamic, and it's changing all the time. My definition is using any type of protected structure to modify the growing environment to maximize production of a crop. So this could be netting, plastic, it could be glass, and we're seeing all of those things being implemented on these farms. So here's all the different types of controlled environments that I work in. But what is different between them is the variability of environmental control. So we go from, there's a technology continuum that we are experiencing. On the low end of the technology spectrum, we have these really simple low tunnels and high tunnels. These are essentially plastic over agricultural land. So you're still growing in the ground, but you're just providing some type of protected structure. And then on the high end here, we have our greenhouses, which are much higher technology. They're very expensive. You don't grow in the ground. You grow hydroponically or in pots. So you can grow on areas that are not set aside for agriculture by using greenhouses. So there is a wide continuum. And on the higher end here, we're seeing more development on the technology side of things to produce plants in really weird environments like warehouses, containers, in space. So there's a lot of investment in all the ponics, hydroponics, aeroponics, aquaponics, and lighting. So all these new LED lights are making lighting more accessible and cheaper by giving plants the exact doses of what they need to be successful. Regardless of the type of system, there are several benefits that apply to all of them. The first one is season extension. So even with a small plastic covering, you can really maximize that growing season. For strawberries in New York, we could extend the growing season by 3 months into the fall. So we were picking strawberries until we had snow on the ground, which was amazing. They can protect against uncertain environments. So by having a structure, you can prevent rainfall, which is the big one, so you keep your plants dry. You can protect against wind and sometimes frost. And what's really cool about these higher tunnels and greenhouses, you can maximize the space within. So this is all kale grown in the winter, but you can see all of the trellis, same for tomatoes. So you can grow up and use that structure to grow up and out. So you can really maximize the space that you use. And with all these things combined, you can really preserve your resources, so land, nutrients, and water. So there's tons of benefits of controlled environments. There are many challenges. The biggest one is the investment of these structures. So they range in how costly they are, but it is an extra investment by a grower. And finding labor who know how to work in these systems is also an extra challenge. So if you build high tunnels, your labor force needs to understand how to use them, how to manipulate them. That's an extra burden. For greenhouses mostly, the energy cost is really, really high and sometimes offsets the benefits. So maintaining light conditions and maintaining a constant temperature is really costly. Pest management is also a challenge. We have some historical and great resources for greenhouse and open field production. For these weird in-between environments like high tunnels, which are basically open to the environment with plastic on top, we have very few tools that are tailored to this because we get a lot of pests, we get more pests in these high tunnels, but the environment is different. So we don't really know how to wrangle these pests. The big three that I encounter in high tunnels are aphids, thrips, and spider mites, and that also applies to greenhouses. But what we're realizing is that research is really lagging demand. A lot of growers are throwing up these tunnels and they're having pest issues and they're coming to us to see what they can and can't use. So we really need to focus on some IPM solutions that are sustainable for the weird types of controlled environments that we are seeing popping up all over the country. And controlled environments have been here historically, and they're increasing in adoption across the U.S. We're actually quite behind globally in terms of our investment in controlled environments. Currently, New York is fourth in terms of CEA value, and we're seeing huge investments by venture capitalists. They're dumping tons of money into the technology side of things to grow plants indoors in cities. As we know, we're all becoming more urbanized. So we need to really think about how we're going to transform some of our traditional agricultural systems into something that can be retrofitted to human environments. So we're seeing a lot of investment here, mostly on the technology side, that we're hoping will trickle down to the applied side. So as an entomologist, I'm more of an applied ecologist. I really think about IPM, and to think IPM, you have to think about the applied ecological questions. So when I'm working on a farm, I'm always thinking about what is determining the occurrence of this pest, what is releasing them from a natural enemy or providing them the right environment to really cause an issue compared to other farms. So I'm thinking about this population ecology type question. So for example, I work a lot with spider mites. They're a huge issue, cosmopolitan pest. And this is a very simple tritrophic interaction system food web we have here. So we do know that host plants can have some benefits or may negatively impact these spider mites. For example, there's a strawberry that has like little trichomes on it. They don't like hairy plants, so you could potentially mix your strawberry cultivars together. And they also experience top-down control by natural enemies. And in an ag system, this is mostly done by predatory mites. So I'm thinking about these food webs. And also, I love tritrophic interaction, so the impacts of these host plants on the trophic levels above. So how are these different types of cultivars or these monocultures or polycultures influencing the sustainability of these natural enemies in the environment? Thinking a little more broadly, how are insect communities assembled? And I'm really thinking about filters, especially in controlled environments, because we do see some things that come in and some things that are not allowed in. For example, we put a sheet of plastic on top of a system. These plastics are UV filtering to varying degrees. So you're changing the light spectra inside of the structure. That has an impact on insects and mites that are very visually oriented. So that might be an ecosystem filter that we might be experiencing. And even within these structures, we are augmenting light. So we are adding in different wavelengths of LEDs, pink, blue, red, sometimes ultraviolet, infrared. That is also changing our insects and what they perceive. We know pretty well how these LED lighting systems are impacting our plants. So what exactly does a spinach need to produce and provide the right chemical profiles. We know what these lighting systems are doing to the plant. We have no idea how that is translating into the insects and some of the direct impacts these lighting systems are having on insects. And also, something I haven't worked with yet, but artificial growing media. So greenhouses are really fun because you can grow in anything. There's a lot of hydroponics, a lot of aeroponics, soilless media. So there's a lot of opportunity to look at these interactions outside of this kind of traditional tritrophic system to see what exogenous forces are pressuring our insect and plant systems. So for the goal of my talk today is to describe some of the tritrophic interactions that I've experienced across the CA systems, ranging from a PhD up to upcoming research that I have planned at Cornell. And I want to pepper in some examples of these exogenous forces that I haven't really thought about before, but now I'm starting to think more about as we engineer these agricultural systems. So low-tunnel strawberry, this is an increasingly popular system in the Northeast United States, so New Jersey included. These are low-cost controlled environments that are adapted for low-growing plants like strawberries or lettuces. It's essentially plastic over ag land. And there's a lot of benefits of these low tunnels. You can extend the growing season of day-neutral cultivars. You can provide some plant protection, but we're definitely seeing more growers invest in these low tunnel systems as kind of a proxy for scaling up into high tunnels. The first goal of my PhD when I started was to describe the community of insects that are present in these tunnels compared to the traditional open fields. I was also interested in mixing up different types of plastics to see if you filter out UV radiation, what happens. I was interested in the broad diversity, not just insects, but we were looking at pathogens and also weed establishment to see, from a grower's perspective, what you'd expect if you are growing under low tunnels. So we used a combination of active and passive sampling to see what was going on in these little systems. And we installed a couple of different plastic treatments. One was a Dubois standard plastic. This is what growers are mostly purchasing. It's very UV transparent, so it's letting in most of that light. And we also found a plastic that is meant more for high tunnels, but we cut it specifically for low tunnels, and this blocks all of UV radiation. So it's a different type of plastic completely. These are the dominant pests that we saw: fruit rots, foliar pathogens like common leaf spot, and some of your traditional subjects that you would expect to see in a strawberry system. We did see, between the plastics, we didn't see any difference for pests. So it didn't really matter if UV was filtered or not for pests, but we did see very clear differences between the open fields and under a tunnel, regardless of the type of plastic that we saw. The tunnels reduced most of the diseases that we saw. We didn't see very much botrytis or common leaf spot. We also saw less spotted-winged Drosophila infestation under low tunnels. And it didn't matter if the tunnels blocked UV or not. So we think this is just a simple physical barrier that's preventing the flies from getting into the fruit. And the UV spectra that's there didn't really matter much. So we do think that this plastic's providing a physical barrier against spotted-winged Drosophila. Some neutral impacts: slugs didn't really care if it was low tunnels or not, same with weeds and thrips. Another one that was a neutral impact was tarnished plant bug over here, Lygus lineolaris, but this was the dominant pest that we saw across all of our strawberries. It didn't matter if there was a tunnel or not. This was a very damaging pest. I think they wiped out like half of our strawberry crops. We didn't apply control for them, so they were a massive issue in our strawberries. One pest that responded very positively to the tunnels were two-spotted spider mites, Chechnicus urticae. This was not surprising. This is a greenhouse pest. It likes hot environments. It likes dry environments. So we were not surprised to see high spot spider mite infestations under tunnels. So these are the two target pests for the rest of my PhD to work on: two-spotted spider mites and Lygus lineolaris. So the second half of this, how do these low tunnels affect the beneficials that we see? The major pollinators were identified. Syrphid flies were a really dominant one. They were out earlier in the season compared to all the bees. The bees, we saw a mix of bumblebees and solitary bees and honeybees. The only effect we saw was for syrphid flies, which is interesting. So the syrphid flies did not like the UV blocking films. And I thought that was a really cool observation because these are very visually oriented insects, right? Like they look at you while you're looking at them. So it's possible that by filtering this UV radiation, they can't navigate inside of these systems, which is interesting that I want to follow up with later on. The major predators we identified were ground beetles, carabids, and miscellaneous arachnids, mostly ground spiders. And they were at a higher density under low tunnels, regardless of the type of tunnel. So they really like this warm and welcoming environment as well. Parasitoids, very similar effect. There was one exception. There was one Chausa group that didn't like the tunnels, but it was a smaller group. But across the board, most of the parasitoids, there was no difference or there was a higher density under tunnels compared to the open fields. So when I was wrapping up my PhD, or this part of my PhD, this is kind of where I was at. So these low tunnels are an excellent environment for growing strawberries. I would highly recommend them. There's a lot of great yield coming out of them. There's no disease, great strawberry fruit. These tunnels don't provide a massive impact for beneficials. There's a couple of exceptions, syrphid flies being a dominant one that I'd like to kind of follow up on. But there are two major pests of concern. One is tarnished plant bug that applies to strawberries regardless of if they're under tunnels, and then of course two spotted spider mites. If you grow under tunnels or in controlled environments, this is a pest that you should keep an eye on. So for tarnished plant bug, I'll call it TPB going forward, or Lygus. This is a cosmopolitan pest. It is present all over. It provides direct damage to strawberry fruits, which makes it a really problematic pest. So it causes this cat-facing damage by feeding on the achenes of the fruit. So it is very tricky. It feeds on all sorts of plants, a lot of weeds in the environment, so it's a landscape level pest that can fly in. We were really interested in microbial control of tarnished plant bugs, specifically using Beauveria bassiana, and one product that we wanted to use was Microtrol. That's a very popular one. We found that it does provide some control of tarnished plant bug in cotton in the southeastern United States. So we were interested in adapting this type of spray to our weird low tunnel systems. The major problem with using this product and other biopesticides is that they're very vulnerable to sunlight. They degrade very, very quickly. So you do not apply them at noon on a sunny day. They will die within an hour. So this is a major limitation for growers, is when to apply it. You have to apply on a cloudy day or at night, and that could be an issue. So yeah, what basically happens, sunlight emits UVB radiation. It hits these really tiny spores, causes DNA damage. This is what it does to us. It gives us skin cancer, right? So these spores, they're so tiny, so that damage is very severe. So our question was, can we actually use these UV filtering films, UV selective films, and apply this product underneath them, and could we expect better control? And you don't have to apply it at night or on a cloudy day because the plastic filters out that UVB radiation. So the summary of this paper, so we applied our Microtrol under a couple different treatments. We had our open field, our traditional low tunnel system, Dubois, which lets in most of that light, and then this blocking plastic. And we evaluated first spore germination, so we washed those leaves and played them to see which ones would germinate. And this is the qualitative result that I like showing, because you can see that the warps plastic is covered in Beauveria bassiana. The other two treatments, the open field and Dubois, a couple of colony forming units, but not very many. So we did prove or provide evidence for this hypothesis that this UV blocking plastic does protect these spores, which we could expect this fungus to perform better under these UV blocking films. And the second step of this is to test this on tarnished plant bugs. So we put out sentinel insects, we sprayed them, we brought them back to the lab and monitored their survival, and we saw more support for our hypothesis. The insects under this warp plastics, they died at a much faster rate than the other two treatments, so we can confirm increased mortality under these UV blocking films compared to a standard film or the open fields, which was great. So our conclusion with this project was that this product performed best under UV blocking films. We do think this is very generalizable because all these biopesticides are pretty sensitive to sunlight, so we think this could be applied to other products containing living microbial biopesticides. So the conclusion of this part is these plastics are an important ecological filter. They are allowing certain things in, they're excluding some other things. They are changing the microbial environment. So there's a lot of dynamics that are explored in these very simple systems, and plastics is one option that a grower can make that can really change the type of environment that they're dealing with. So we want to think of plastics as an IPM tool, depending on what the grower would like to see in their fields. If you're interested in plastics, we did produce a video on UV selective films and how that could impact plants and insects and other things. So you can go there, it's on the Tunnelberries YouTube channel. So the second pest that we were dealing with are two-spotted spider mites, Tetronychus eutychi, or TSSM. This is a worldwide pest of everything. Almost all crops are susceptible to this spider mite. They really love hot and dry environments, so they are a huge greenhouse pest. If you buy plants from Lowe's, check them, because those palms have spider mites. I have dealt with this many, many times. I never learned because I like these palms. But yeah, they love controlled environments, indoor plants. This is that food web that I showed you earlier. We were interested in evaluating the different trophic levels and combining things to see what would happen So we published some work on host plant selection by spider mites, which cultivars are better or worse and why. We looked in some biocontrol options, comparing different species of predatory mites and mixing them together. And then we did one last project on tritrophic interactions, and that's what I'm going to focus on for this part of my PhD. Biocontrol by spider mites, of spider mites by phytoseeds is a classic and old system. Spider mites are a major target of the augmentative biocontrol industry, and 60% of the predators that are reared and sold are accounted for by 4 predatory mite species. And these predatory mites range in their diet preferences. There are some, like Amblysseus horskeyi, that will feed on pollen. They'll feed on thrips, whiteflies, some broad mite species, and also spider mites. So they range in their diet preferences, but they're a huge player in the augmentative biocontrol industry globally. The one predator that is the most popular among them, that is a specific predator for spider mites, is Phytocellulus persimilis, this red individual here. This predator is a specialist of the Tritrophic genus, so it will feed on red mites and two-spotted spider mites. It's not native to the United States, so we release it anyways, but there's no expectation that it will overwinter, except for maybe in a greenhouse or in a high tunnel. Growers perceive this as a prophylactic biopesticide for spider mites, so there's no expectation that it will linger for the rest of the season. They'll go in, eat down your spider mites, and you'll have to re-release these predators once they come back, because they're not going to persist because they're a specialist predator. And a lot of greenhouse growers, they just release these weekly, regardless of how many spider mites they have, even if they have no spider mites, because it's cheaper for them just to get weekly shipments and to release them to prevent a spider mite outbreak. We released these in the summer, a couple of summers in New York on our strawberries, and they didn't work. We saw very, very low recovery, which is really curious because this is an old system. It should work, and it just wasn't. So I wanted to kind of push this a little further. And my question was, these commercially reared predators, are they poorly adapted to this environment I'm throwing them into? Like, are they just not set up for success based on how they're reared? So a little bit about the rearing processes for these predatory mites. This is changing, but this is kind of a historical perspective. They're traditionally reared on these high-density bean plants. It's a very simple rearing system that are full of spider mite prey. So these predators have a very easy plant to navigate, and they have lots of abundant prey that's easily accessible. When prey starts to run down, they naturally disperse, and this is how they're collected, and they're shipped off to be sold and released. So they climb up these nets into these collecting vessels that are packaged and sold. And then you release them. Yes, they climb up the netting. And then we release them on the back end, and we put them on all sorts of different crops in the field. And there's this expectation that they'll work. And based on this rearing system, they're adapted to this one plant, lots of prey. So you put them in this very challenging environment on something like strawberry that has glandular and non-glandular trichomes that they have to deal with. The prey might be lower quality or scattered. So these predators have a challenge that they must overcome in the field. So I had this question of how this host plant rearing system is impacting their dispersal and their movement on a challenging crop in the field. So this is my hypothesis 1. So we have these predators that are adapted to this bean environment. You release them onto a weird plant, right, that they've never been exposed to. They don't like it. And in response, they disperse. It's an unacceptable host plant. They hit the plant, not compatible. They disperse, and that is why we're not recovering them like we think we should. Our second hypothesis is that same thing, we have these predators, we release them onto a plant. They're not dispersing per se, but they have the inability to locate and feed on prey. So they might be sticking to the plants because they're trying to navigate those trichomes, but they can't find prey. They're starving, and that's why they're not working, and that's why we're not recovering them. So we have these two hypotheses that we were weighing. And the way we compared this is we had 2 different colonies. We first got our predatory mites from a distributor. We put them on a standard commercial bean colony and also on a strawberry plant that had spider mites, and we reared them for at least 5 generations. Then we put both of them onto a strawberry plant to see how well they can navigate that system. And we measured movement and dispersal in a couple of ways. We wanted to scale this up. So the first experiment was a lab kind of interleaf disc arena where we put 2 leaf discs next to each other, and we put a staple right in the center to see if these predators can cross that bridge to find a prey patch on the other side. And the way we track the movement of this, so we put predators on one end, we put prey on the other. We wanted to see if they could find that prey spot. and we tracked their movement using this tracking software called EthoVision to see when they crossed that bridge and how far they would walk to find their prey. In the second experiment, we scaled it up to an interconnected plant experiment in the greenhouse where we had strawberries, we put prey on one end, we released predators on the other. We wanted to know how many of them could cross that artificial bridge to find prey. And the last experiment was in the field under low tunnels. We inoculated strawberries with spider mites. We put predatory mites in there, and we wanted to see how far they could walk and disperse in a low tunnel field experiment. For the first experiment, this was so fun. EthoVision is a really great software. It works so well for mice and bigger organisms. For tiny things, it really sucks, but we were able to figure it out. So this little dot is the predatory mite, and the software is picking it up most of the time. And it's rounding around, it's confused. Eventually it finds the staple, thinks about it. No. Goes back. Goes back again. Can't decide. Eventually pops over, and when it's rounding the circle or the edge like that, it's looking for a place to disperse, so it's not liking where it is. Eventually it settles down and starts feeding on some prey eggs. But what's really cool...
That's a strawberry leaf?
Yes.
Okay.
Mm-hmm. Yeah, so the software works pretty well. It picks it up most of the time. This is a very powerful software, so you don't have to sit and watch them. You can actually just let it, like set it, forget it, and then the data you get back is so cool. You know how far they've walked, you know where they spent their time, you can see how many times they've crossed that bridge. So you can get a really great data set on behavior using EthoVision, but it's really hard to do with a really small organism. We tried this for spotted wing, by the way, Drosophila. It picks it up perfectly. So spotted wing Drosophila, it works really well. You can talk to Greg about that. So the data on this is the proportion of individuals that were able to cross that bridge and find prey. The two treatments are divided by color, so the blue will be bean, the red will be strawberry. They're both observed on strawberry. And we did see that the strawberry-reared individuals had a much easier time navigating that strawberry leaf disc and finding prey on the other side. The individuals reared on bean had a more difficult time. They weren't very successful in their interleaf dispersal. Which gives more support to our second hypothesis that it's not that they're trying to disperse from the plant, they're having a difficulty navigating it to find what they need, their prey. When we scale this up to the whole plant experiment, we didn't see much of an effect. This was over in the greenhouse. We didn't have EthoVision. It doesn't work for a whole plant experiment. I wish it did, but it'd be magic if it did. This was over a 24-hour period, so it's possible. It was just enough time for all of them to make it over, but we missed some important interval. Maybe the strawberry reared individuals got there first, and then the bean ones followed, so maybe we should have done this at like 6 and 12 hours instead of a full day. So yeah, we didn't see much. Most of them were able to cross, well, about 30% of them were able to cross, and that was the same for both treatments. So we didn't see much of a difference here. In the field, we repeated this 3 different times. We put 150 individuals on a center plant, and around those 2 plants, we identified plant 1 and plant 2 that were just right next to them. We bridged them using the plant runners, so they did have a bridge between the plants. And we did it on both sides of the center plant. So the 30 meters and centimeters are actually 2 plants around the center plant, if that makes sense, by combining them. And you might be wondering, okay, these predatory mites are identical. How do you know if they didn't like bleed into each other? Well, we thought about that. And we developed a cool marking system using this really funky, weird product called Smart Water. It's a crime scene, weird spray that you can mark things with. So if you live in a town where your bike gets stolen, you can mark it with this Smart Water. If you have a UV pen, it will shine. So you can like write your name on it, and it's invisible to us, except under UV light, and you can see it. What's cool about this product is it comes in different colors. So we can mark predators with different colors, so we can discern colonies. So we published this. It's really great. If you have a great system where you can basically mist these predatory mites, the droplets are fine enough that they don't drown, but you can pick them up under a UV pen. So we really liked this little study we did. Okay, so for recovery, recovery is pretty low but not bad. But we did recover more bean reared, that commercial strain, on the center plant compared to the strawberry reared individuals. And that held up over all of our distances. So our result is that there's more support for the second hypothesis. The strawberry reared individuals are dispersing more so than the ones that were reared on bean. So the bean reared individuals are sticking to the plants a little bit better, but they might not be performing very well. So we have support for this hypothesis that we have this commercial strain, we put them on a strawberry, they're actually not dispersing very well. But the second important half of this is, okay, does this lead to poor prey finding and eventual starvation? Is that why we're not seeing them perform very well in the field? So we evaluate this directly. Our strawberry colony tend to be more dispersive on we tested them on beans and different host plants. Strawberry colonies were more dispersive than the bean colony, just across the board. So we put both these colonies on a strawberry plant infested with spider mites just to see how well they could eat down those spider mites compared to a control. This is the number of spider mite prey eggs over time and split by our different treatments. We have a control, obviously without predators, they go out of control. And then the bean colony didn't do as well as the strawberry colony in terms of eating down those prey. So the strawberry colony seems to be better adapted to this plant, which it is because it was raised on strawberries, and is resulting in better biocontrol. So maybe that's because they're more dispersive. So just the simple fact that they can walk around better on the plant, they can find prey and they're encountering prey. Or there's another hypothesis is that the strawberry root individuals, yes, they walk around more, but they can also better find these target prey, they can handle them better. Spider mites root on strawberries are a lower quality than those root on bean. So these strawberry adapted individuals know how to handle this prey, they can digest it better, they can convert it into energy a little bit more efficiently. So there's another kind of a physiological response that we are not getting at all, but that could be leading into this result as well. So at the end of this project, what do I say to the commercial rearing industry? Because they don't want to increase the complexity of their growing systems, right? So based on my study, it might not be a bad idea to include some complexity into your rearing system. So your predators have encountered these weird plants before, they can perform better in the field. That is not an answer that they want to hear, right? They're trying to go to completely plantless rearing systems for predatory mites, even specialist predatory mites. They don't want any plants in there at all. It's messy, hard to rear, more risk for contamination. So that is an unlikely adoptable piece of advice, I guess, that they would consider. However, what I can say is that release rates should really reflect the environment that these predators are released into. So we know that they're not going to be as adapted to strawberry. We need to release more of them on a strawberry plant compared to a bean plant. Tomatoes, again, they're even worse than strawberries. There are lots of trichomes, they're sticky. So if you have a spider mite issue, you need to release double the amount on a tomato plant to get the same benefit. And currently the release rates don't really reflect this. So we need a more individual recommendation based on the level of spider mite infestation and the type of plant that you're releasing them on. So that is the best recommendation I can give to the commercial industry and to growers. So that was finishing up my PhD work. So now we're scaling up into high tunnels. When I was finishing up my PhD, I knew I wanted to go into controlled environment something. I loved working in tunnels and low tunnels. It's just drier, it's nicer. You can work in the winter. So I really liked these environments. So I found a postdoc that was working in these high tunnel systems. So I joined Dr. Laura Inglow's lab working on high tunnel pest management as part of a larger SCRI. So this is the team that we had. So you might recognize the entomologists on the left-hand side. We had some horticulturalists in the center, so Becky Seinemann and Wenjing Guan. And then we had our ag economists on the group as well. So whatever recommendations that we had or results, we gave them all of our data so they can say, okay, this maybe works better, but it's really expensive, so that's not likely to be adoptable. Whereas this thing worked not bad, but it was super cheap. That is going to be more adoptable. So it's important to include the economists on your research because they can really help push it over the finish line. Okay, the objectives of our SCRI, which I'm no longer on, but I'm kind of a part of still. We were really interested in monoculturing versus polyculturing under a tunnel system. So tomato is like the classic summer system. And in the summer, tomatoes are still there, but some growers throw in a bunch of weird plants in there as well. So how do these different systems impact pest pressure? Also, how does it compare across the urban gradient? So a tomato grower in a rural area, would they experience the same level of pest pressure compared to a tomato grower in an urban area? Again, those ecosystem filters, what are allowing some things in? And then also, we really need to think about four-season production. These tunnels are used all year. So we can't just be thinking about our classic summer tomato system. We should be thinking about winter as well, which is what I will focus on for the rest of this talk. So we were really interested in describing the high tunnel community. This happened in October 2022, and we are still collecting data on this. There are 3 researchers across the United States working on this. I was the lead for Indiana, Purdue, and we have Gary McCauley, who is a PhD student working in Illinois, then Anna Wallingford up in the northeastern New England area. We visited 40 farms across 3 states every month for 2 years. And we collected data on the types of insects that we saw. And we're still collecting data and still organizing it. So I'll just present the data that I collected from Indiana because I have that data with me. We chose sites that varied along this landscape gradient. We are kind of qualitatively calling these urban and rural sites. We have a partner who is actually doing all the GIS mapping to really give these a more fine definition, what an urban site is compared to a rural site, because it's also very squishy. So we will be kind of maybe changing this a little bit, the definitions here. This is what our growers tended to look like. So they're all very unique and different. No 2 tunnels were the same. We had some growers that really took the polyculture to the extreme, and this is like a wild garden, basically. Then we have some growers that have a monoculture, really neat and tidy crop. Then we have some in between. We have some DIY, customized high tunnels, and then more standard tunnels that are on the bottom right-hand side. That's what we typically see. But they were all over the place, and the types of crops that they grew, how they managed their crop. So super, super diverse group. We visited each farm, and we first looked at the crops to see what types of natural enemies and pests were present. And the primary pest that we saw in the winter and the dominant crop that we saw across the farms was spinach. So that's the crop that I will focus on. Aphids were the dominant winter crop on spinach. We think this is a mixed kind of species. We do think that there are some overwintering potato aphids that are coming off the tomato that were grown in the summer that are jumping onto the spinach. We also see green peach aphid and a couple others. But they are active in the winter and they are causing problems. Pill bugs, you might be puzzled by this. These are detritivores. They should be great in the soil until you have too many of them. And a lot of our tunnel growers go to the extreme in terms of organic matter and having a really great soil complex. They have a lot of rich soil that attracts a lot of pill bugs, and these pill bugs do feed on the plants. So radishes, anything close to the ground, these guys will eat. So they were actually a pest in my systems. Fungus gnats, obviously. Ants with a question mark, because we don't know if they're causing issues with the aphids, or if the association or not, but they're active during the winter. Thrips and then flea beetles at the really tail end of the winter season. But yeah, aphids and pill bugs were my biggest concern. For natural enemies, we did see some parasitized aphids. So we think that is the dominant natural enemy, that's attract the aphids that are there. Lots of spiders are active, and Casey Athey, my collaborator at Illinois, is a spider person. So we're looking into the species of spiders, and she is doing a lot of gut content analyses to see what they're actually eating. Because spiders are cannibalistic, they're opportunistic feeders, so they might be eating each other or other biocontrol agents, or they might be eating the pests, we don't really know, but they're active in high numbers in these tunnels. And we also saw adult parasitoids flying around, don't know what the species are, some ground beetles, amphicorids, and then some syrphid fly and cecile fly larvae that were active as well. So winter management of aphids. So we know it's a massive issue. What are we going to do about it? There's not a lot of recommendations for winter management. There's an assumption that it's too dark, it's too cold, nothing's going to work. And also growers aren't aware that there's an active pest problem happening in the really dark, cold days of winter. But these aphids really capitalize on a couple of warm afternoons, and they will reproduce. So a lot of growers will not do anything until it's too late. So they put their crop in, they put a cover on top. Couple weeks later, they uncover that, and it's just aphid city. And so my question was, what IPM options would be effective in these wintry conditions? I always like to go back to this IPM triangle. I really like this because I can kind of pick and see which growers would use which things and which things can synergize together. So I really love biocontrol, so I started there. So we had 12 high tunnels to work with, which is wonderful, but a ton of work. We put spinach in these high tunnels, and we inoculated them with these potato aphids that we think are the bridge species from summer to winter. And then each tunnel we assigned to one of these 4 treatments. So we had 3 tunnels that we didn't put any predators out. 3 tunnels were released Adalia bipunctata, that's the 2-spotted lady beetle, Aureus insidiosus, and then green lacewings, Carsoperla carnea. And this is the impacts that we saw. And I'll present this as the pre-count versus the end of season count, so we can see that level of increase. So we did start off with kind of naturally occurring higher aphid densities for the green lacewing treatments just by happenstance. At the end of the season, by May, The aphids in the no predator treatment went out of control, right? We had lots of aphids per spinach leaf, beyond a threshold, I would say. For all the predator treatments, they all reduced that rate of increase. They actually all did a pretty good job in maintaining lower levels of aphids. One treatment in particular, the green lacewings, actually provided curative management. So it took care of that higher density that we saw before. And these happen to be the cheapest ones. So we highly recommend green lacewings in winter conditions for aphid management. So all of them worked pretty well. They vary in how cost efficient they are, but I do think that green lacewings are probably the best bet if you want to save money. Okay, so I can't just think about biocontrol by itself. There's a lot of interacting factors that affect the sustainability and the efficacy of biocontrol. So I was looking across this kind of avoidance part of the triangle, and I was really interested in cultural control, specifically weed management. My master's degree was in weed biocontrol, so I love weeds. And also landscape effects, right? Landscape effects, kind of the effect of urbanization on pests and natural enemies. So starting with the weed management, whenever we visited these farms, we did a quadrat assay to determine how weedy it was. And we gave it a score of 1 to 5 based on percent weed coverage of these quadrats. These are all the farms that I visited in Indiana. We did see that there is a positive relationship between weediness and pests per spinach. But we also saw the same effect for natural enemies. So as you increase in weed density, you should expect more pests, but also more beneficials, because they're probably just following that prey source. But what we notice is that not all weeds are the same in terms of the types of insects that they attract. Some weeds, like henbit, hosted tons of potato aphids. So if you're growing tomato in the summer, you have lots of aphids, you have this weed that's popping up around fall. If you don't take care of it, those aphids climb over to the weeds while you are intercropping with something else, and they will walk back over to the spinach plant. So this weed in particular is a bridge for these potato aphids. There's also some weeds where we saw a lot of molds, white molds, that could impact some of the brassicas that we were seeing. So weeds, are a likely bridge for some pests. However, not all of them are. We did see some weeds that had actually more predators than pests. A lot of the clovers that we saw were harboring a lot of green lacewing eggs, and they didn't have a lot of pests on them. So we think that there's some weeds that are actually not completely terrible. They actually recruit some natural enemies, which is wonderful. I'm not likely to recommend growers to leave weeds around. They're not going to do that. But from an ecological standpoint, I see these as a companion plant system. Like, they do attract some pests, but they attract natural enemies, and hopefully those things balance out and keep pests away from your crop. So I have a broader perspective of this, but right now I'm not advocating towards keeping weeds around. But a lot of folks are interested in keeping weedy strips because it's easier, no maintenance. And they do provide some benefits over kind of a cover crop or companion plant system that you actually have to maintain. So there's some fodder here, but it's unresolved. So stay tuned on that paper. That'll come out hopefully soon. For the landscape effects on biocontrol, I was really interested in urbanization and how it affects these winter insect communities. Here's one farm that's in downtown Indianapolis, has a high tunnel. They struggle greatly with aphids, and I was wondering, why do they have an aphid problem and why don't our other growers do? They're not doing anything much different. So we looked at the insect counts on our spinach plants, and the way I'll divide this is by our kind of qualitative assignments of rural, suburban, and urban, and we gave these assignments kind of based on this farm is in downtown Indy, that's our urban site. This one's a little bit further away, so we gave it a different category as suburban. So they might change, but we think this is pretty solid for now. So we'll have the percent of counts on spinach, and we'll divide it by the feeding guild. So the first group are the herbivores, or mostly aphids. We do see that urban sites have a higher percentage of herbivores or pests on their farm compared to the suburban and rural sites. Interesting, right? The next group are the predators. So we see more predators in the suburban sites, which is really cool. Not very many in the urban sites. That could be potentially contributing to the higher aphid numbers. And then parasitoids. We saw very, very few parasitoids at our urban sites. None. The suburban sites, again, had the highest percentage of parasitoids at their farms. And just the other groups, just smattering of other things. More detritivores in the rural sites compared to the urban sites, so this includes those pill bugs. And then more ants in the urban sites, which might be because of the aphids. So there is this kind of urban community happening. So we do think that parasitoids are one major group that is being limited in these urban environments. So we do think that there is an effect of urbanization on our pest communities and the ability of natural enemies to persist on an urban farm compared to a suburban or rural farm. So the conclusion of my postdoc work for this side of things, we do think that aphids are the dominant pest of interest on winter crops. We do think that some weeds may be a bridge for pests to build, so careful weed management should be done. Augmented biocontrol using commercially available natural enemies is an effective IPM option, and it's compatible with other strategies. We do think that parasitoids are a dominant resident natural enemy, and it's being limited in urban environments, leading to higher pest outbreaks on urban farms. We do think that for parasitoids, there is an option or an opportunity for conservation biocontrol on urban farms. We do know from our previous work in community gardens, Indiana, at these urban sites, when you have a higher crop density or higher crop diversity, we have higher insect diversity. So this is a very strong positive relationship, which is completely intuitive, right? You have a diverse crop system, you should have diverse insects, it makes sense. But we do think we can apply this to our high tunnel systems. But I'm really interested in these other sources of plant diversity. I love weeds, and I wish that we could appreciate them a little bit more. So I do see this, even though a grower is very good about managing their crop, I see these weedy barriers all the time. So you can't get to these margins. It's really hard to till that or to get a weed eater in there. So there's these naturally occurring weedy barriers. I want to know what role these weeds are playing in the environment compared to a companion plant system. So I have a couple questions here. Can we consider weeds a beneficial source of plant diversity? How do they compare with companion plants that are put there intentionally to conserve our natural enemies and pollinators? So this is the last study I did. I'm still crunching data on this. We established different treatments of high tunnels. We had a monoculture tomato plot. We added some companion plants into another treatment. And then for the last treatment, I allowed in natural weeds. But the way I did this, you can't just have a weedy tunnel that's unmanaged. So in the plastic mulch, I just put holes where I would put companion plants. I let the natural weeds come up. So we standardized the number of plants in the high tunnel so we can make more direct comparisons. Our objective was to compare the efficacy of these companion plants that are there intentionally versus these naturally occurring weeds in terms of enhancing biocontrol and pollinators. This is what our sweet alyssum looked like. This is such a popular bedding plant used on farms and tunnels. It is so attractive to all sorts of things. Everything likes this plant. Every area you can see things crawling around. This is a huge risk for thrips though, so it's great for some things, but for thrips, they love this plant. So there are trade-offs here. And I'm not quite ready to get into the details of this project yet because I'm writing it up currently. But based on the community matrices that we put together, they are very different in terms of the community of insects that we see. The weeds are so different than companion plants. Even within the companion plants, there are differences between the herbs, so the basil and the dill that we would consider companion plant if you let them flower, are more closely together. The marigolds and the alyssum are actually quite different, which was interesting to me because I thought they would be more similar than the others because they're a flowering plant, they produce a lot of pollen and nectar, so I thought they'd be more closely assembled. So yeah, all these points represent the community matrix. If they're closer together, they're more closely related. So it's neat and nice that the weeds cluster together, so those treatments were the same, and so that's how this map kind of works. So yeah, kind of interesting division happening amongst our treatments. So we're going to dig into that a little bit more and determine what the risk factors are for a weedy tunnel versus tunnels with companion plants. So yeah, there's some interesting stuff happening here. There's a ton of work that we're doing. We are year 3 or 4 of this SCRI, so we're just getting our data together. We're about to publish a bunch of stuff. We have a website, so if you're interested in high tunnel production, you can visit that and you can see all the cool stuff that we're working on. So I think I am, yeah, perfect, rounding the corner here. So these are some experiments that I have planned, I'm proposing to do in my new position at Cornell Agritech. I'm an insect ecologist and I'm working in a controlled environment, so I can work on any type of crop or pest or pollinators group that I want to, but it has to be within contained systems, and I get to define what that is. So I can work in tunnels, I can work in greenhouses or low tunnels, so that's pretty fun. The first project I'm going to kick off this spring, optimizing biopesticide sprays in greenhouses. I'm really interested in working with these entomopathogens, and they would work better in a greenhouse than they would in the open field, hands down across the board. So we really should be using these biopesticides in greenhouses. I'm really interested in that vertical canopy because it gets so hot in a greenhouse as you go up, and these are vertically trellised crops. So if you apply this product on the canopy to target a specific pest, will the product actually work up top, whether it's closer to the light system, it's hotter up there compared to spraying mid-canopy or the below canopy, because there's different pests and they assemble themselves differently. So I'm interested in that, and also comparing the endophytic activity of these pathogens. So maybe they don't do well on the topical surface or as a topical spray, that they're better able to infect and colonize the plant tissue, so they can become endophytes and provide some protection against some chewing insects that might be coming around. So that's something I'm kicking off this spring. My second experiment, I just got funding for this, disentangling the effects of LED lighting on pests between the indirect and direct effects. I mentioned before that we know how light impacts plant chemistry and morphology. We don't know how that translates up into the pests. So a spinach that's grown in infrared light, is it more protected, less protected against aphids? But also, how do these lights impact the aphids directly? So if you grow them up in these different lighting systems, are they bigger, are they smaller, do they have a lower reproductive rate? So what should we expect in these greenhouse systems? We're really playing around with light. And also, same for the natural enemies. How do these light systems impact their behavior and their biology? And the last project is continuing this work on companion plants. I want to know what makes a good companion plant. You want something that is attractive to beneficials, not attractive to pests, and can we find an ideal system? And if we do, can we actually provide some artificial sources? I'm really interested in these grasses. I see a lot of lacewing eggs on grasses, so that could just be a structural component. It's a nice erect grass, and it's really strong, and lacewings like to oviposit on that. If that's the case, we could provide that artificially without introducing more plant material. I'll preface this by saying a lot of our greenhouse growers that grow spinach and tomato, they don't want companion plants. They don't want to bring in more diversity. They want a clean system. To get into the greenhouses, you have to wear like a hazmat suit because it's clean. So they're not going to be into bringing in grasses or flowers because that's a risk for them. So if we can find some interesting qualities of the plants, could we artificially provide it some way? Maybe make some type of, I don't know, bushy material that's not plant but maybe made of cotton to preserve some of our biocontrol agents that we release in there. So I'm interested in that. I'm interested in comparing the physical, nutritional, and chemical profiles of companion plants and really comparing them and how they support the beneficial insect community in controlled environments. That is all I have for you today, and I think I'm right out of time. I'm growing my lab. I just started, so if any of you are interested in joining my lab in the future, please contact me. That's my email at Cornell. You can follow our group on Instagram and follow me on Twitter. Yeah.